Hello, everyone. Olá a todos. I will now share my screen. Okay, and let's start. I am Roberto Refinetti from the University of New Orleans. Sou Roberto Refinetti, da Universidade de Nova Orleans. And the title of my talk is Animal Models for the Study of Circadian Rhythms. I have a few notes in Portuguese at the bottom of the screen. All right, so let me start by pointing out or reminding everyone why we do medical research, right? Medical research is conducted to improve human health, but experimentation with human subjects is greatly limited by ethical concerns. Humans don't like to be poked with needles, don't like to be stuck in a room in the dark for a month, don't like to have their food messed up with. So research is conducted on other animal species, as well as in plants and other life forms. Circadian rhythms have been studied in a large number of plants, animals, fungi, bacteria, and other life forms. I myself have studied rhythms in 37 different species of animals, including ants, lizards, birds, rodents, dogs, sheep, horses, and camels. Now, the most used organisms globally by other researchers have been, among the plants, the mustard plant, Arabidopsis thaliana. Among fungi, bread mold, Neurospora crassa. Among invertebrate animals, the fruit fly, Drosophila melanogaster. And among vertebrate animals, the domestic or laboratory mouse, Mus musculus. So again, these were the 4 species mostly used very widely among circadian biologists. Each of these model organisms has several advantages and several disadvantages for research. A major advantage of vertebrate animals, of course, is their similarity to humans, especially if they are simian primates. And here is a marmoset monkey. Those of you in the audience, most of you in Brazil, know very well the marmoset monkey. This is not a fully grown marmoset monkey, but almost. And as you can see here, this is a coffee mug. And he's barely taller than a coffee mug. It's a very small monkey, very convenient for studies, especially in a laboratory. Now, on the other hand, the fruit fly does not look much like humans, but is very good for genetic and molecular research. Actually, studies with fruit flies in the late 20th century and beginning of this century led to the award of the 2017 Nobel Prize in Physiology to 3 circadian biologists. So fruit flies have a long history in the study of circadian rhythms. Now, most researchers using mammalian animals consider that rodents provide the best compromise between convenience and similarity to humans. Again, rodents are not as similar to humans as monkeys are, but they're much easier to have in a laboratory, are easier to breed, are easier to maintain, resistant to diseases. They're very good animals. A main consideration in the choice of a species is whether the rodent species is nocturnal, like most rodents are, or diurnal, like humans are, whether they are active during the night or they're active during the day. This decision is not as easy as it sounds, for the simple fact that diurnality is not an all or none matter. So let me start by showing you some of my own data here. For a number of laboratory rodents, there is a gradient of diurnality, not a sharp division. So this is a study published back in 2006 in the Journal of Comparative Physiology, showing some rodents, and I'll talk a little more about them later. And some are grouped and the thing in here, but we have at the very top here, the grass rat. Let me see, yeah, here it is. The grass rat, Nile grass rat, which is very much diurnal. So sorry, what we're showing here on the x-axis is a diurnality index, which is simply the quotient of activity during the light phase of the light dark cycle divided by the total activity. So if the animal is active exclusively during the light, it will be a perfect 1. That's diurnal. If the animal is exclusively active during the dark, it will be a 0, it will be nocturnal. And actually, as you can see, the Syrian or golden hamster, there's a picture there, is very much that way. It's a truly nocturnal animal. The grass rat, yeah, it's almost fully diurnal, not quite. I'll show you more data about diurnal animal rodents later. This is what I had at that point. And you see that there's a gradient. It varies. Some species are more diurnal, others are more nocturnal. If you wanted to use 0.5 as a separation, you would have 3 diurnal animals here. And 1, 2, 3, 4, 5, 6 nocturnal animals in here. But again, why an arbitrary cut when there is a continuum from one end to the other? Now, in a larger number of mammalian species, the means of the daily onsets of activity also show a dispersion, but is not as radical as in the diurnality index. This is a study conducted 10 years later with a much larger number of animals. As you can see, we're going from body mass of a few grams to more than to 1,000 kilograms. Horses and cows in here. These are very small mice and other rodents in here, some squirrels, rabbits, cats, dogs, sheep. Anyway, I guess the most obvious thing in this graph is that body size doesn't tell much whether you're diurnal or nocturnal, except maybe there are a few more diurnal animals on the high end here. But in the lower end of body masses, let's say under 10 kilograms, there are some animals that are diurnal, some animals that are nocturnal. Now, interesting here, we're showing here, so is the daily onset, when they start to be active. And as you see, they're kind of close in here, not perfectly on the line, with lights came on at 7 o'clock and off at 7 in the evening at 1900 hours. These animals are pretty close, some a little before, some a little after the lights come on. And this other group in here, you know, starting pretty much around the lights when they're going off. So there's still some variability, but not as much. Let me emphasize that. So animals are reasonably differentiated into those that start activity close to sunrise. I guess you could call diurnal, and those that start activity close to sunset that you could call nocturnal. But they showed, as I showed in the previous slide, 2 slides ago, they show a gradient in the time when they are most active. Also, some animals have rhythms that are very clear, whereas others distribute their activity almost equally throughout the day. The term robustness is used to refer to the strength of rhythmicity, so how close to a smooth sine wave. And robustness varies greatly from species to species. So you should see here again, I'm showing with body size, it looks like maybe robustness is a little higher on the lower body size, but that's not the main point in here, is how it varies so much from, you know, very low. It's almost like it's not rhythmic. I don't know, 5% in here to over 50%. Now, why not 100%? Well, this is general activity. There are some things you will see more. I don't have time to get into that detail in this talk, but when you look at even just basic running wheel activity, the robustness is greater. Body temperature, which I'm not showing here at all, is usually much higher 70, 80, 90%. So it's a very nice and clear daily rhythm. In any case, again, the strength of rhythmicity varies from one species to another. So even if you could separate between diurnal and you can't really separate between diurnal and nocturnal. That's the main point. It's not a very easy way to do it. Additionally, although the means of activity onsets for each species seem to be well defined, and you could, I guess, call them diurnal, nocturnal, the inter-individual variability, the chronotype spread, varies greatly from species to species. So let me show you this. Again, this is from the 2016 study. Sheep at the top here, cat at the bottom, very precise. So chronotype spread. So that's where 95% of all individuals in that group start their activity. And as you see, sheep are very precise. Almost every sheep starts at the day at the same time. Cats, if you have a cat or if you have more than one cat, you know, cats can be up during the day, can be up during the night. They might wake up in the middle of the day, in the middle of the night. Very relaxed thing. So the variation between one individual and the other is great. In the case of the cats, probably within the individual, there's a great variability too, but let's not get into that right now. So you see a very big difference. Now, interesting in here is that, oh, there we go. There's some rodents are very similar to humans. So here's the human. Human is about 6 hours. So everybody wakes up in the almost 95% of people wake up somewhere between 5 o'clock and 11 o'clock or 6 o'clock and noon. Most people wake up in that window. And as you see here, this squirrel is very similar, the laboratory rat, Nile grass rat, Siberian hamster, they're very similar to humans. Not all. The Syrian hamster is down here, Dagu further away. But anyway, some of the rodents are very similar to humans in that respect. Okay, so several rodent species that are reportedly diurnal in the field have been tested in the laboratory and found not to be reliably diurnal. So I'm going to mention them briefly. I'm not going to go into detail about them. These species include the golden spiny mouse, the fat sand rat. In this picture, it does look fat, and it's called fat because it's a little chubby. And the tucutuco down in South America, that, you know, it's diurnal, and you can see this picture taken during the day when it was out of the hole underground. And in the lab, they don't turn out to be too reliably diurnal. But there have been 4 rodent species used as diurnal models for the study of circadian rhythms with some regularity. So I would like to talk about them. My research collaborator and I examined these 4 species in greater detail. And this is the article published in Laboratory Animals in 2018. My collaborator is Professor Refinetti in Washington. One of the species is the Mongolian gerbil, right? Myriones unguiculatus. It's a smaller animal, not small as a mouse, but 90 grams. They're originally from China and Mongolia. They're a common laboratory animal. They are available from, you know, breeders for laboratory animals. About 50% are diurnal. The other 50% are nocturnal. So anyway, the other species is the beagle, Octodon degus, which is much larger, body mass at 240 grams, is almost the size of a rat. So he's a smaller, younger rat. They're indigenous to Chile. They're an exotic pet, at least in the United States. You can find, not everywhere, but in some pet stores, you can find babies. They are diurnal, but they have strong crepuscular surges during twilights. Anyway, that's the dagu. Another one is, another of these four species is the Nile grass rat, Arphicanthus niloticus. The body mass 120 grams, so bigger than a mouse, also bigger than a gerbil, but not as big as a rat. Well, young, very young rat, 120 grams. Anyway, they're indigenous to North Africa. They're not available commercially. Currently, I know there are at least 2 labs that have them. Well, at Michigan State University, Professors Nunez and Yan have the Nile grass rat. And Andy Gall at Hope College also has Nile grass rats. And the fourth species of those 4 is the white-tailed antelope squirrel, Amospermophilus leucurus. It's called antelope squirrel because the tail looks like an antelope tail turning over like this. Body size is the same as the Nile grass rat, a very convenient 120 grams. They are indigenous to North America. So people doing research in North America to be able to go and catch them. They're not available commercially. You'll have to go and trap them in the wild. Currently, I know of 2 labs that have them. My lab at the University of New Orleans and Professor De La Iglesia lab at the University of Washington. Okay, so the basic experimental setup to study these 4 species was this. The animals were individually housed with a running wheel, the running wheel inside their cage. They could jump on the wheel and run anytime they wanted. They were maintained at 24 degrees Celsius and their LiDAR cycle with 12 hours of light per day, the light being 360 lux. And they had food and water ad libitum. Okay, so let's see then first some representative actograms of running wheel activity for the 4 species. And as you can see here, the antelope ground squirrel, very nice. So this is an actogram. I assume everybody knows what an actogram is. You have 1 day per line, each day under the other. And so you can see the one, and here shows the light. So the light comes on at 8 o'clock in the morning, it goes off at 8 o'clock at night for 12 hours of light per day. As soon as the light comes on, maybe slightly later, the animal wakes up, jumps on the wheel and runs pretty much the whole day, and stops right when the light is going off. The Nile grass rat, here called African grass rat, also runs very regularly during the day. Starts usually, these are typical animals. Of course, not all animals are the same. There's some variability, but generally the Nile grass rats start running a little before the lights come on, and they stop around the time and the lights go off. The Mongolian gerbil, again, 50% of them are nocturnal. This is a typically diurnal one. Does start running when the lights come on. They're not very regular throughout the running period, and they might go away into the night with some activity. The dagu also starts somewhere around the lights on, but continues later, and you can see, a pause in between here, and not as regular as the antelope ground squirrel, for instance. And I'm sorry, so there's some comments. That was an excellent record, a good record for the Nile grass rat, not good for Mongolian gerbil, good but kind of too wide for the dagu. And that study published in 2018, Refinetti and Kennedy. All right. So analysis of many of these parameters of rhythmicity, I'll show you in a second, indicated that the antelope squirrel has the strongest, most robust, and most consistent pattern of diurnal activity of the 4 species. So here are the data, and we're showing a number of things in here. This is the diurnality index, and so the antelope ground squirrel is the first one in red. The Nile grass rat is the second one in orange. Mongolian gerbil is this other orange. And the dagu is in yellow here. And as you see, diurnality is the highest in the antelope squirrel. Acrophasis is when it peaks a little after lights on for all of them. They're definitely decent diurnal animals in that sense. The onset of activity, when they start around the lights on, a little later for the gerbil and a little later for the dagu. Alpha, I guess at this point here doesn't matter too much. That's the duration of the activity in a day. And there isn't very much of a clear difference in them. Robustness, how strong the rhythm is. And it's very strong in the antelope ground squirrel, not as strong in the others. And finally, the distance that they run each day. Of course, it depends on what you provide them with, but wheels are different sizes and different things. An antelope ground squirrel runs about, on average here, 8 kilometers each day. For many of you, or many of us humans, running 8 kilometers a day would be great exercise. We wouldn't, be like in the United States, more than half of the population now is overweight. If we ran 8 kilometers a day, we wouldn't be overweight. And we're way bigger than these guys. So 8 kilometers for them is probably 20 or more kilometers for us. Anyway, let's move on. Oh, one more aspect in here. The free running period of the antelope squirrel is the same as that of humans, the average being 24.2 hours. So the internal biological clock runs at the same speed in the antelope ground squirrel as it does in humans. The other animals vary. Some of them are actually shorter than 24 hours, some are longer. Okay, because the antelope squirrels seem to be the best animal model for human chronobiology, we use it to study the determinants of chronotype variation. There is chronotype variation in humans, right? Everybody heard about that, with extremes being called morning types and evening types on the other end. But the causes of the variation are not known and cannot be studied with the rigor of animal research. Again, people don't like to be stuck in a room and kept there for weeks with their food controlled, light controlled, and everything else. Variation in human chronotypes has been extensively studied. I'm going to just briefly show data from my own laboratory. So these are from 207 university students. They were given the morning as evening as questionnaire. That study was published in 2017. And you have the evening types. In this case, the score is lower score means evening, higher scores mean morning. And we didn't get the typical normal, almost normal distribution that you get in a very large population. But then again, college students tend to be a little on the evening side, and that's what we got, a little more to the evening side than in the middle. All right. Now here, let me show you a much larger group of people from 5 countries. That's not the morning evening questionnaire. This is just the activity counts. But I have this information, this study we published in 2015 with more than 400 people in each country. So we have more than 2,000 people in here. And that's the average distribution of when they're of activity. So as you see, activity peaks. This is in Ghana, peaks at around noon, 12.3 o'clock. In other countries, it's a little later. I arranged them in order of later. In the United States, the peak activity is at 3 o'clock in the afternoon, as opposed to noon in Ghana. The light dark cycles with the time when we conducted the data collection were pretty much similar. So that doesn't explain the difference in the time of the peak, but that clearly reflects something about chronotypes that people in the US like to stay, be more active later in the day than in some of the other countries. All right, so to study the determinants of chronotype with an animal model, we use the antelope squirrel. And here is Molly, an antelope squirrel eating an almond. If you don't think this is the cutest, let me run again. Animal you've seen. So they really enjoy almonds as a treat. It's not their main diet. All right, so what did we do? That's, again, data study that we published in 2019 in Physiology and Behavior. These are two extreme cases. This just to show you that we do have a morning type and an evening type in the squirrels. So this animal woke up around sunrise or lights on and went to sleep actually slightly before lights went off. This one started much later. This is what, 1, 2, 3, 4 o'clock in the afternoon. I understand we have about 300 people in the audience, so I'm sure there's at least one person who feels like this squirrel. But again, the range in humans, it's in this area here where people wake up between 5 and 6 and 12 o'clock. But there are some people who are very late types, and this was a squirrel that was really late. And you can see it starts running in the light. Then, of course, the light go off, it can't run as well. So you see a little noisier thing. And actually ends up not running as much during the day as that guy did, because it doesn't really like to run in the dark. But that's what his clock is setting it to. Right? That's what chronotype is all about. That's what this study is all about. What is determining? Why is this animal starting activity so much later than this other one from the same species? Actually collected in the same, pretty much the same area. So what were the findings when we looked at all these parameters of rhythmicity? Well, chronotypes were stable attributes of individual animals. So when I tested, let's say, those two animals again, 6 months later, the correlation in their times of onset of activity was 0.91, very good correlation. So it is a stable attribute of that individual. Chronotype is a characteristic of the individual. In squirrels as it is in people. But again, in people, you can't really do that. You can't put them in the dark and test them and then test them, well, you can test them 6 months later asking, but you can't really test them. You have to ask them. And some people, yeah, you can put them, give them some Fitbit or some other activity monitor, but you can't really control their environment to make sure that, it didn't change 6 months later. Anyway, another finding is that chronotypes were significantly correlated with circadian period and with alpha. Alpha is the duration of the activity in each day. We don't have time now to go into this, but that helps explain why, or at least it's a potential explanation of the differences in chronotype, because a shorter circadian period would require the animal to start activity earlier or a longer period. Anyway, the change, a difference in circadian period affects how much light you need to get each day, and therefore when you need to start getting light. So that could determine the phase of entrainment, is to call the chronotype of the animal. Another finding was that chronotypes were not significantly correlated with photic sensitivity, which again, we don't have the time to go into, but that would be an important determinant of where you're gonna, your relationship with the light-dark cycle, because your sensitivity is gonna determine either parametric or non-parametric, meaning depending on the mechanism of masking or entrainment by pulses. And that's what we did here, masking of activity or phase shifts caused by light pulses. And those were not related. So animals that shifted more did not have earlier or later chronotype. So apparently it's mostly on period than on the phase shifting ability. or the masking of it. And also, chronotypes were not significantly correlated with putative personality traits. There aren't standard personality trait tests for squirrels or any other animal as there are for people. We designed our own tests, so you could argue that those were not really measuring the personality traits. Those are the big 5 personality traits in humans. And we tried to measure those with some tests, and we did not find correlation with chronotype. So presumably that's also the case in humans, but of course, that's something to be determined later. Finally, I must acknowledge that having a good physiological model, as our squirrel seems to be, may not be enough. Right? Biomedical research using animal models very often requires knowledge of the genome of the species. And only 2 rodent species have had their genomes sequenced: the mouse and the rat. Importantly, they're both nocturnal, so they won't be of much help if you're trying to study a diurnal animal. So, the take-home message from this presentation: animal models are very helpful in medical research, including research on biological rhythms. A major advantage of mammalian models is their similarity to humans. Rats and mice have been used extensively, and their genomes have been sequenced. But rats and mice are nocturnal, whereas diurnal rodents are more similar to humans in terms of circadian organization. 4 rodent species have been used as diurnal models for the study of circadian rhythms with some regularity, and they were the Mongolian gerbils. They are easily available, but are not very diurnal. Degus are relatively available, but are not strongly diurnal. Nile grass rats are not easily available, but are consistently diurnal. And antelope squirrels must be trapped in North American deserts, but are very strongly diurnal. So let me thank you for your attention, and I'll be glad to answer questions now. Perguntas podem ser feitas em inglês ou português. Let me get out of the screen and stop sharing my screen.